The California Technology Council's new CTC Benefits Trust combines groups of emerging technology companies to offer large company benefits to small businesses. This approach delivers employee benefit programs with better choices and at a lower cost. What's included? Medical, dental, and vision options are available, with additional employer and employee online resources to support simplified enrollment and administration. To learn more, go to californiatechnology.org/join. That's californiatechnology.org/join. I'm Daniel Levine, and this is the Bio Report. While much of the focus of drug developers looking to act on the genetic mechanisms underlying diseases have focused on the genes that code for proteins, Syros Pharmaceuticals is looking to the regulatory regions of the genome. This is the non-coding part of the genome that controls the expression of genes, turning them on or off, or modulating their level of activity. We spoke to Eric Olson, Chief Scientific Officer of Syros Pharmaceuticals, about the company's discovery platform, its pursuit of cancer and monogenic diseases, and the lead programs in its pipeline. Eric, thanks for joining us.
Happy to be here.
We're going to talk about Syros Pharmaceuticals, its approach to drug development, and its pipeline. We did speak to your CEO, Nancy Simonian, in early 2018, so we're going to cover some new territory here. Syros is developing therapies that target the regulatory portion of the genome to turn gene expression on or off, or to modulate it. Perhaps we can start there. What makes up the regulatory portion of the genome?
Yeah, so the genome is divided up into actual genes. There are about 20,000 genes in the 3 billion base pairs of the genome, and that's really a very, very small percentage of kind of the landscape of the genome. And the rest of it, for many years, people have been trying to figure out what is the rest of this DNA and what is it doing. At one time, it was even called junk DNA a couple of decades ago. But what we now know is that a large part of that genome is really there to direct the transcriptional machinery, or the machinery that drives the expression of genes in a very cell type and context specific manner. It's really those parts of the genome that direct all that transcriptional machinery to the right genes to get expressed at the right time in the right cell.
How well understood is the role of this part of the genome and the role it plays both in health and disease?
Yeah, well, first of all, the recognition that it plays a very important role, that's very clear. We know that many of the most important elements that drive the expression of the genes are in these regions of the genome that you actually can't identify just by sequencing the DNA. You need other methods. And really, the last 10 years, have seen a revolution in the kinds of methods that we can use to dissect and understand what these regions are, what genes they're regulating, and the role they play. Now, in terms of disease, there's also a lot of work over the last several years by geneticists looking at what we call GWAS, or kind of gene variants in the genome that are associated with certain diseases or predictive of worse outcomes in diseases. Well, it's turned out over the last several years that many of these variants are exactly in these regions, these regulatory regions of the genome. So every month there's new data making these links between genetic variants and and the genes they control and how they play a role in disease. And in fact, in monogenic diseases like sickle cell disease, for example, we know that one of the therapeutic opportunities there that we may have a chance to talk about is to use the understanding of that regulatory region around the globin locus to turn on a new globin gene.
Well, before we talk about any specific indications, how do you go about targeting this portion of the genome to interfere with the process underlying a given disease?
Yeah, so first of all, you have to understand, obviously, what those regions are, what are they doing, and what parts of the machinery are they influencing to control the expression of that gene. So you kind of got to get the parts list of what are all the parts that are interacting with that region of the genome to drive that biology. And then you have to basically go into the lab and start manipulating those parts, see which ones really play a major role and might offer an opportunity to be a target for a specific drug.
Well, take a step back and talk about how you go about target discovery and preclinical development. Do you think you're doing anything fundamentally different than other companies to identify the candidates you're pursuing?
Yeah, we have three parts to what we're doing. First is an understanding of what this transcriptional machinery is in a various cell type associated with a specific disease, and kind of its interaction with the regulatory regions around those genes. Second is kind of deep biology of understanding that disease and what is the intersection of the pathology and what the patient is actually experiencing and what's happening in the patient's cells with regard to that. And then the third is drug discovery capabilities to actually find the drugs that can influence that biology. So we start with defining the problem at the gene control level, making sure that where we speculate to intervene, we really convince ourselves that that's going to have an effect on the disease, and then we go about building the drug discovery platform needed for that particular drug. But we always start with patient tissue, patient cells, and really try and characterize this regulatory landscape directly in the patient tissues instead of model systems. You know, for many years we did drug discovery taking findings in models, whether they're the fruit flies or mice or rats, and doing deep characterization there, and then looking for drugs in those systems, and then, in essence, going out and hoping we find patients that look like the model. We kind of start just the other way around. We say what's really happening in the patient's cells in the disease state, and then we actually build our models based on that understanding.
You're pursuing both cancer and monogenic diseases. You alluded to sickle cell a moment ago. The approach makes sense to me in the case of something like sickle cell disease, where there's a single faulty gene underlying a pathogenic process. Cancer, though, tends to be a lot more complex than the aberrant activity of a single gene. What's the case for using this approach in cancer?
Yeah, one of the things that we know happens for a normal cell to become a cancer cell, it really has to, we call it rewire its genome. It really has to change several aspects of the transcriptional program in order to become a tumor cell. So one way to attack that is, for example, find a drug that would, what we call pleiotropically, affect the ability of that cancer to kind of run that transcription or that gene control program. And we can talk about our two of our programs where we're applying that. But the basic concept is with a single drug, you're going to be influencing the expression of many genes. And in fact, that will lead to the death of the cancer cell.
Is there a risk, though, of having an off-target effect in broadly acting that way?
Yeah, so that's one of the reasons we look at specifically in starting with tumor cells from patients and compare them to normal cells and ask, what is the transcriptional misregulation, and where can we intervene where we can disrupt that misregulation in the tumor cell with minimal disruption in the normal cell. And it's really only understanding both states, the tumor state and the normal state, can we find drugs that will preferentially kill the tumor cells. And the tumor cell will reveal several what we call dependencies or transcriptional dependencies. For example, many oncogenes, or genes that are really important for the cancer, have to be produced at a very high level continuously, where that's not the case in a normal cell. And if you lower or shut down the expression of one of those really critical oncogenes, the cells can't tolerate that and go into a process called apoptosis. Whereas normal cells, they're not as sensitive to that because they don't have this heavy dependency for constant high-level expression of those genes.
We're seeing a growing movement toward combination therapies to treat cancer. Is there a case to make for using what you're developing in combination with other therapeutics? Are you looking at these as single agents?
Yeah, absolutely. And it is a growing movement to use combinations, and it makes perfect biological sense. Since usually by the time we treat people with cancer, that cancer could be very heterogeneous in terms of the types of cells that are in there. They're not every cell is exactly the same as the other cell. So one rationale for combination is different drugs will kill different subsets of those tumor cells. Sometimes we can build on, for example, in some cancers, they've deleted a specific gene, and they're reliant on the other copy, on the other chromosome. So they only have a single copy. So they're already crippled in terms of the level of expression of that gene, where the normal cell still has both copies. So if you knock down that one in the cancer cell, again, the cancer cell can't tolerate that because they only have one copy. And based on whatever pathway that is in, we can look to other drugs in that pathway for synergistic effects. And we're in our current trial, we're exploring combinations with both our RAR alpha agonist, SY1425, and our CDK7 inhibitor, 1365. We have arms in the trial where we are treating in combination with agents.
When we had Nancy on the show, we discussed your lead candidate, 1425, which is being developed for AML. I'd like to focus on your other clinical candidate, SY1365, but can you give us a quick reminder of what 1425 is and where you're at in development there?
Yeah. So SY1425 is a molecule that acts on a transcriptional regulator protein called RAR alpha. And in fact, in a subset of patients with AML, this RAR alpha is misregulated, and it in turn then misregulates many other genes. So here's an example where we can modulate the activity of RAR alpha and have a downstream effect on the expression of many genes. So that's basically what the molecule is and the pathway. So we have in a phase 2 trial with SY1425 in combination with a molecule called azacitidine in what we call biomarker-selected AML patients. These are patients where through our platform and understanding this regulatory region of the genome, we saw that this RAR alpha pathway was misregulated. So in that collection of patients, we're looking in both relapse refractory AML patients, as well as patients who are newly diagnosed AML but are unfit for standard chemotherapy. So both arms are enrolling, both here in the U.S. And in Europe, and we're looking forward to that data.
SY1365 is a CDK7 inhibitor. You're pursuing this both for ovarian and breast cancer. It's not a new target of interest within the industry, but it's been a difficult one. What's the interest in this target, and what's been the challenge in getting drugs to work on it?
Yeah, the interest in the target really comes from some work. First kind of arose from some work done at Dana-Farber Cancer Research Center at Harvard, and with one of our co-founders, working with various cancer researchers, had discovered that many cancers have this transcriptional dependency where they need to express these high levels of these oncogenes, and that CDK7 inhibition was a really potent way to shut down the expression of these genes. So that came through some work through academic labs, and so we decided to make a molecule. Now the challenge there was there's a whole family of proteins called CDKs, and to get a very selective CDK7 inhibitor with a small molecule was not trivial, because the site on the protein where you want the molecule to fit is very closely related to the other CDKs. So the challenge was making a selective molecule that hits CDK7, and then of course had all the rest of the drug-like properties that you need to take a drug into the clinic.
And how does the drug actually work?
So SY1365 works by binding into a pocket of the CDK7 protein, but it also does has this mechanism which we call covalent inhibition, where the molecule basically permanently attaches itself to CDK7, and in essence kind of cripples the CDK7 protein. It keeps it from working. Now, CDK7, the other interest in CDK7 was, I told you about the transcriptional, what we call the transcriptional dependencies or misregulation. Well, it turns out tumor cells also are misregulated in what this process called the cell cycle. Normally a cell, there's various checkpoints for a cell as it's through its life, where it's making a decision, should it replicate its DNA and divide or not. Well, cancer cells have kind of blown through that decision process and keep replicating. And it turns out that CDK7 is an important protein in driving cells through the cell cycle. So we're actually, by inhibiting CDK7, we're really attacking two types of biology that the cancer cell is misregulated and needs to survive: cell cycle and transcription.
You've got a second CDK7 inhibitor in preclinical development. How does this differ from 1365?
Yeah, so 1365 was the first CDK7 inhibitor in the clinic, and it's an IV molecule. As I said, it permanently attaches itself to CDK7. While we were discovering and developing that, we were working on a second molecule that would be oral. We know there's going to be many other cancers where this mechanism could be a valuable mechanism, but where an IV therapy might not be the ideal route for a patient. So we set out to also make an oral molecule. So SY5609 is our second CDK7 inhibitor that we're preparing to take into the clinic early next year. And it's highly selective, potent molecule that we're excited about.
And do you know what indications you expect to pursue?
Yeah, we're currently We're currently testing the molecule in several different types of cancers, and throughout this year, and that data will really be the data that will help us decide where we're going to take that. Of course, we're learning a lot about SY1365 and ovarian cancer and breast cancer, so we'll certainly be building on that. But again, given that it's an oral molecule, kind of the breadth of the cancers we could go into is probably a little larger.
Eric Olson, Chief Scientific Officer of Syros Pharmaceuticals. Eric, thanks so much for your time today. Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.